All right, so we are still in the week of January 25th through 28th. We're finishing up chemical level of organization. Hopefully we can start cellular level of organization today or Thursday, okay? So we should have a mastering A&P quiz sometime this week to do. All right, so next week is exam one, all right? Be over chapters 1, 2, and 3. So just wanted you to be aware of that. All right. So let's go ahead and talk about lipids. We talked about carbohydrates last time. Remember, these are types of organic molecules. So lipids are mostly hydrophobic. Remember, what does phobia mean? Fear, doesn't like, right? Doesn't like water. These are things like fats, oils, and waxes, and made up predominantly of carbon and hydrogen atoms, okay? So types of lipids, you have fatty acids, you have eicosanoids, glycerides, steroids, phospholipids, and glycolipids. And again, don't write all this down because again, I just pulled this straight from the chapter, right? So I want to make sure you're mainly listening for kind of the big points, okay? All right, so let's start with the fatty acids. So these are long chains, have a lot of carbon and hydrogen in them, and they have what we call a carboxyl group at one end. And a carboxyl group is carbon, 2 oxygens, and a hydrogen, COOH. All right, it's a relatively non-polar, right? Because it's just mostly carbon and hydrogen, right? Remember, those bonds are generally non-polar covalent bonds, except for that carboxyl group, which is going to be very polar. Fatty acids are said to be saturated or unsaturated. Okay, anybody ever seen at the grocery store when you're getting the butter, it says saturated fat or unsaturated fat? Anybody seen that before? Yes or no? Maybe you don't go grocery shopping, I guess. But so to be saturated means you're saturated with hydrogen atoms. Okay, it's like wet, like it's just full of, like you're just saturated with hydrogen atoms, which means you're not going to have any double bonds in that tail that's made up of hydrogen and carbon, which is what we call a hydrocarbon. So to be unsaturated means you have at least one, if not more, double bonds in that hydrocarbon tail. If you have one double bond, you said that fatty acid is monounsaturated. If it has multiple double bonds, it's polyunsaturated, okay? So we'll show you kind of the implications of that. All right, so here's lauric acid. So with lauric acid, you see this long tail here. This is a hydrocarbon tail because it's hydrogens down here, okay, and up here, and a bunch of carbons. So it's hydrocarbon. Does that make sense? All right, so don't get caught up in, oh, there's a bunch of letters and whatever, just a bunch of carbons and hydrogens, right? So that's nonpolar. But then you have this carboxyl group, COOH. Okay. Oxygens tend to make very polar covalent bonds. Okay, so this part's polar, the rest of it's nonpolar. All right. Here's the saturated versus unsaturated. So if it's saturated with hydrogens, and here they didn't draw all the hydrogens, okay? But you just have a bunch of single bonds in the carbon tail, or in the carbon portion of it. If it's unsaturated, you have at least one double bond, okay? And the implication of that is it changes the shape of the fatty acid, okay? It's no longer just this, rigid pattern. Now you've got a kink in the pattern, okay? Now, that's actually a good thing. Why is it good? Because if you're eating a bunch of, pizza and buttery, get some rolls with some butter and all that good stuff, all that fatty acids, then once it starts to get in your your circulatory system after you've been through your digestive system, starts going around in your blood vessels, right? It's floating around in your body. If it's all saturated, if it all looks like this, they can just stack right on top of each other. Okay, because it's kind of like, you know, if you get chairs and they're all the same chair and you can just stack them on top of each other, they just fit on each other, right? Same kind of thing. And that's bad because if they stack on each other, eventually they're going to get heavy and sit somewhere in your blood vessel and just stack and stack and stack. And what does that do to your blood pressure? Goes up, right? You get atherosclerosis, arteriosclerosis, high blood pressure. So you don't want that, right? You don't want it clogging up your arteries. The unsaturated fats are better for you because it makes it harder for them to stack on each other. Does that make sense? Okay, that's why they're healthier. All right. Now we talk about eicosanoids. So funny name, but that's just what they're called. So these actually can't be synthesized in your body. Some things you can make. Your body can make certain things, but these you have to get from your diet. Okay, you gotta eat them essentially, or get them in some sort of pill or something like that. Nutritional supplement. They're derived, or they come from a fatty acid called arachidonic acid. Okay, so that's their base, like what they use that to make these. All right, and examples of these eicosanoids are leukotrienes. And anytime you see leuco, it's typically going to be like a leukocyte. You know, these are all things that have to do with the immune system. All right, so they're active in the immune system. And then you also have prostaglandins. And these are fatty acids that have short chains. Okay, those nonpolar hydrocarbon tails are short. And they act as hormones in a localized area. Okay, which means they're chemicals that have some effect. That's basically what a hormone is. All right, here's an example of a prostaglandin. Okay, I'm not necessarily caught up in you having to know a whole bunch of chemistry or organic chemistry. What I'm really interested in is you understanding this is a hydrocarbon tail. Okay, because it has a bunch of what in it? What letters? Carbon and hydrogen. Okay, same thing here, another hydrocarbon tail. Okay, so when you see those, you're probably looking at some sort of lipid. Okay, if you see long hydrocarbon tails. All right, and that's a short one, because remember the prostaglandins are short. The other lipids are much longer than that. All right, the glycerides. And this is sort of a compound where you take a glycerol molecule, which we'll show you in a minute, and then you attach fatty acids on top of it, okay? Now, if it's glycerol and one fatty acid, it's a monoglyceride. If glycerol and two fatty acids, it's a diglyceride. Glycerol and three fatty acids, it's a what? Triglyceride, okay? And the triglycerides have other names. Sometimes you'll see neutral fats. Sometimes you'll see triacylglycerols. Okay, so what are they good for? Well, they're an important energy source for your body. And they also provide insulation and protection. Okay, that should make sense. I mean, you think about bears, right? And they eat a lot and hibernate. Why do they get all that fat, right? Because A, they have to sustain their body for energy while they're sleeping. B, they have to insulate themselves because they're usually someplace that's really cold, right? So, and fat is essentially a shock absorber. It's a protector. All right. Again, you know, don't get freaked out by this. Okay, this is glycerol, and before you again get freaked out by glycerol, really all it is 3 carbon atoms, some hydrogens, and 3 OH groups. And you see, OH is alcohol. And that's why it says glycerol. So, you know, most people think when they say alcohol, they're talking about ethanol, but we're talking about the category. All right. So anytime you see OL, it has these OH groups. Okay. So in here, we're going to go through dehydration synthesis, right? Because we're going to remove water and make a new compound with the new bond. And that fatty acid chain is going to attach to glycerol. Okay. These are saturated because they have single bonds. This one's unsaturated because it has a double bond. Thank you. All right. So if you do dehydration synthesis, you get the triacylglyceride and the water's being removed. If you do hydrolysis, you can break it up into the individual components. Okay. Right. Monomers, polymer. Right. Remember we talked about that a little bit. Polymer, many, mono, individual units, one. Okay. So again, I'm not asking you to draw any of this stuff. I will have a question or two where you see some structures and you kind of have to generally be able to tell me what it is. Okay. Again, I'm not asking you to tell me, well, that's this specific chemical. I'm just asking you to tell me a category. Okay. All right. Then we have the steroids. So the steroids they have this characteristic structure. They have 4 rings, okay? If you see a structure that has carbon atoms arranged in 4 rings, okay, it doesn't really matter what the other functional groups are. If you see the 4 rings, it's a steroid, okay? A classic example of a steroid is cholesterol. And where do you see a lot of cholesterol? In your plasma membranes in your cells, surrounding your cells. And again, before you get freaked out and say, wait a minute, how is cholesterol a steroid? I thought steroids were for making people muscle bound. That's one type of steroid. We're talking about a category. A lot of times in TV, they take a category and make it the name for something. All of those steroids, like the steroids that, you know, allegedly Barry Bonds and, you know, everybody was taking in baseball, right? They have specific names, you know, like androstenedione and some of these other ones. But the category is steroid, right? So, and they don't all do what those steroids do. So also your sex hormones like estrogen and progesterone, they're steroids. Why? Because they have that 4 carbon ring structure, right? So steroids like calcitriol, these are hormones, or corticosteroids. So we have some hormones that are steroids. You also have derivatives, or, you know, where you use a steroid as the basis for it, like a bile salt. And bile is part of your, you know, it works in your digestive system, helps you break down things. Okay. So again, category, right? Steroids, a category of lipids. All right. Here's some examples like we just talked about. All right. It's cholesterol. This is estrogen. Okay, so say hi, cholesterol. All right, nobody did it. That's okay. All right, so this is estrogen and this is testosterone. Okay. I'm not going to ask you to know all these little functional groups along here. Okay. I'm not going to ask you to tell me, hey, you know, I'm not asking you to write down this chemical formula for testosterone. Okay. If you want to take organic chemistry and biochemistry, be my guest. You can do that kind of stuff. And it's actually kind of fun, but if that's not what you want to do in life, that's fine. But what do all 3 of these structures have in common? 4 carbon rings. Okay, thank you. Okay, so again, I'm not a detailed person. If you got to know me, I'm a big picture person. I'm not I got to write things down because I forget, right? I'm not a detail, or I mean, I'm detail oriented to the extent I need to be, but I tend to be very, what's the story? What's the big picture? Okay. And so for me, the big picture here is 4 carbon rings is steroid. Okay. That's what you need to know. So lipids. So these are phospholipids and glycolipids. All right, so then this is another category. So we've gone over like the eicosanoids, the steroids, fatty acids. All right, now we're at phospholipids and glycolipids. All right, and these you actually can make in your cells, okay? And they're gonna be composed of a diglyceride. So a diglyceride means it has how many fatty acids attached to the glyceride? All right, very good. And they can be attached to either what we call a phosphate group, which has phosphorus, the element phosphorus in it, or a sugar. If it has a phosphate group attached to it, we call it a phospholipid. If it has a sugar attached to it, we call it a glycolipid. Why? Because glyco means sugar, okay? Root words. All right, generally, both of these are gonna have heads that like water. They're hydrophilic. Remember, philos means loving, and tails that are hydrophobic, which means they don't like water. And here you got an example, you have structural lipids, and these are what you find in your plasma membranes, in your cell membranes. Okay, so this is not a robot who's running across the page. This is actually a phospholipid, okay? So although if that helps you remember, that's fine. So again, don't freak out on me here, okay? Let's break it down into little parts, okay? So, all right. So this is kind of like, you know, Power Rangers or Voltron, if y'all remember that. We had the robot that all the pieces that came together, right? So here are your fatty acid chains, right? Your hydrocarbon tails. Okay, this one is saturated or unsaturated? Saturated, right? No double bonds. This fatty acid chain is unsaturated, right? Because it has a double bond. Okay, it's attached to our good friend glycerol. You know it's glycerol because it has 1, 2, 3 carbon atoms. Okay, you've seen those before. Okay, so don't freak out on me. Attached to a phosphate group. Why? Because it has phosphorus in it. Okay. And then you have some sort of non-lipid group up here. Okay. So it has a different chemical composition, which typically is going to be polar. Okay. Remember, this is the head's going to be polar, likes water. The tail's going to be non-polar, doesn't like water. Okay. So that's what I'm saying. Don't freak out on me. Just look at the pieces. Okay. And just go back to what do I know? And let's start filling in pieces. All right. So this one, we just went over the fatty acids, the glycerol, and here's the non-lipid group. This has these 6-membered rings. 6-membered rings usually, unless it's the 4 rings together, if it's just individual rings, it's a carbohydrate. So if it's a carbohydrate, a sugar, what kind of lipid is this? Just learned it on the other page. Started with the G. Glycolipid. Whereas this one, because it has a phosphate group, it is a phospholipid. Okay. All right, so here's another example. Again, phospholipid because it has phosphorus here. Glycolipid has sugar. These are both hydrophilic, like in water. Okay, so this biological application, you have what we call a micelle, where all the hydrophilic heads are on the outside, the hydrophobic tails are on the inside. Okay, so it forms a little sphere, okay, to kind of get away from water, or at least to get the tails away from water. Okay. So that's lipids. And the thing is, again, we'll talk a lot about the lipids when we get to chapter 3 and talk about the cell membrane. But just want to give you an overview. All right, so what I'd like for you all to do now is to talk with your battle buddy or battle buddies, and each person needs to explain 2 types of lipids, okay? Y'all can do this now, and if you're watching live, you can do this while you're watching at home. So now that we've covered the lipids, let's get to the proteins. So the proteins are arguably the most important organic molecules because they are typically the ones who do the business of the cells. Okay, and they're also the most abundant, which means you see them most often. So typically going to have some basic elements to them that you'll see in every protein: carbon, hydrogen, oxygen, and nitrogen. Okay, CHON, or if you need a word, I mean, it's not really grammatically correct, but you could say honk. Whatever works for you to remember those 4. Okay. Now, remember when we talked about carbohydrates, the monomers, we had the monosaccharides, right? And then you could make polymers like disaccharides and polysaccharides. For proteins, the monomers are amino acids, and there's 20 of them. And you can put them together in different combinations to make different proteins, okay, which are the polymers, okay? So amino acids are the monomers and proteins are the polymers. So what do proteins do? Okay, this is pretty important. I gave it its own slide, right? So you probably need to make sure you pay attention to these. So proteins are involved in providing support. So they provide structural proteins. So some of them are structural and they provide support for the cell. Others are involved in movement, like proteins that are involved in contraction, okay, contractile proteins. Others are involved in transport, like carrier proteins, okay, helping things move from one side of the cell or one inside or outside of the cell. All right. Other proteins are involved as buffers, which means they can help regulate pH, keep it in a certain range. Other proteins are involved in metabolic regulation. Okay, what does that mean? That means they regulate chemical reactions. Okay, and one of the ways that they do that is through enzymes. Okay, we'll learn about those in a few minutes. All right. You have coordination and control, okay, through hormones. Okay, so some hormones are proteins, and they can basically, those are chemical signals that tell your body what to do. And others are involved in defense. Okay, so they provide ways to keep your body safe from foreign invaders, like antibodies. Okay. So support, movement, transport, buffering, metabolic regulation, coordination and control, and defense, right? So those are important functions of proteins. All right, so let's talk about the structure of the proteins. So here you've got these long chains made up of those monomers, the amino acids. So what do the amino acids have in them? So every amino acid is going to have a carbon atom in the middle, okay? So And it's going to have around it a hydrogen atom, an amino group, which is NH2, nitrogen and 2 hydrogens. A carboxyl group, we've seen that before, COOH. And what we call a variable group, or R group. So carbon has 4 bonds. It can make up to 4 bonds. So those are the 4 things that go around the carbon atom. Okay. All right, show you what that looks like. So here's your central carbon atom. Here's your hydrogen atom, your amino group, NH2, your carboxyl group, COOH, and your R group, your variable side chain. Okay, and this is what tells you which amino acid it is, because all this other stuff is consistent, right? We said there were, do you might remember how many amino acids we said there were? 20. So there's 20 different R groups, okay? All right. So to make a protein, you have to link 2 amino acids together. How do we do that? With our old pal dehydration synthesis, right? So here we're creating a bond between the amino group of one amino acid and the carboxyl group of the next amino acid, okay? Forms what we call a peptide bond. And we call it that because the molecule that's created is a peptide. So peptides are polymers, and polypeptides are anything larger than 2 peptides. So it's a tripeptide and larger. Okay. So here are 2 amino acid monomers. You have glycine and alanine. So glycine, its R group, variable side chain is a hydrogen. Alanine's variable side chain is CH3, right? Dehydration synthesis, you lose water, form a bond, you get a peptide bond. And you release water. The reverse reaction is hydrolysis. You use water to break. And then you break into the individual monomers, the amino acids. Okay? But remember, we've seen these 2 chemical processes before, right? We're just using it in the context of proteins, right? So this is the way that this whole course is going to work. You're going to learn things, and then you're going to build on it. You're going to have, every time you learn something new, you're going to have to say, what do I already know? And how does this sync with what I already know? Okay? And it helps you remember it, because then you're building a framework, and your framework just gets bigger and bigger, as opposed to just a million random facts, right? So protein shape. So we talked about the structure. Now let's talk about the shape. And it has basically 4 levels of structure. So we'll start with primary structure, which is just your amino acid sequence, just whatever the list is of the amino acids in the sequence. Okay, that's your primary structure. Then you get your secondary structure. And in the secondary structure, we're actually looking at some folding. And that's through interactions with hydrogen bonds. Remember we learned about those, right? We have polarity, differences in charge create the hydrogen bonds. And you'll see structures like spirals or sheets in that. It's secondary structure. For tertiary structure, you actually get more coiling and folding. And the big thing there, the key word for tertiary is 3-dimensional. Okay, because tertiary is third, and it's 3D, right? 3-dimensional. And quaternary structure, it's found the protein complex that's produced by the interaction of different polypeptide chains. So it's kind of taking your tertiary structure of different chains, add them together. That's your quaternary structure. So primary is your sequence, secondary is your hydrogen bonding, giving you spirals and pleats. Tertiary is your 3D structure. Quaternary is multiple polypeptide chains. All right, so what does that look like? Okay, so here's your linear chain of amino acids. It's your primary structure. But the secondary structure, based on this hydrogen bonding, these interactions in the molecule, you get secondary structure. Okay, in this case, you're forming an alpha helix. Okay, it's a spiral. So in this case, you still have primary structure in the linear chain, but the way the hydrogen bonding works for this molecule, you get kind of a folding. It looks kind of like a folded piece of paper. Okay, so we call that a beta pleated sheet. All right. And again, we're not going to have you draw this. We're not going to have you, make this. It's basically going to ask you, you see a beta pleated sheet, which level of structure is this? And you're going to say secondary. Okay, that's the kind of stuff you want to be able to do. Okay. All right, tertiary structure. It's your three-dimensional structure. Okay, the whole proteins or whole subunit, or your whole polypeptide subunit. This is what your whole thing looks like in 3 dimensions. Okay, you take all the alpha helices, all the other portions, all the folding. Now, here, this is quaternary structure. Why? Because there's multiple protein subunits involved. In this case, we're talking about hemoglobin. Hemoglobin actually has 4 units that work together. And that's important because that allows you to carry your oxygen in your red blood cells. Okay? So, can everybody see the difference between tertiary and quaternary? How this is multiple, this is singular? All right, but if you see the word 3-dimensional, you're probably talking about what level of structure? Tertiary. If you see the words multiple subunits, you're probably talking about which level? Quaternary, right? So that's what I want you to be able to do. Here's another example. So here's your tertiary structure, 3D structure. Here's a quaternary structure. In this case, you have multiple spirals, multiple subunits with spirals working together. So it's like spirals on spirals, okay? All right. More on shape. Sometimes some proteins are considered globular, which is like a sphere. And it's soluble, which means it can dissolve. And the shape is based on the tertiary structure, right? The 3-dimensional structure. You also have fibrous proteins. And these are going to form sheets or strands. Okay, so this is going to be focused on secondary and quaternary. Why? Because sheets are in the secondary, right? And these strands, multiple subunits, is quaternary. Okay. All right. So I think I'm going to pause here, talk about what you know about proteins with your, so each person should say 3 things that they know about proteins. Okay. And then we'll go on to enzymes. And you can do this at home as well. All right, so let's move on to enzymes. So enzymes are one of those functions of enzymes, or one of those functions of proteins we said was metabolic control. And so we're involved in reactions. So enzymes, what they are, they're a class of proteins and they are catalysts. So that's a big word. So I think they're in the study guide, it was one of those sentences that had enzymes are blank, blank, blank. They're catalysts. And what does that mean? They lower the activation energy for a chemical reaction. All right, that's what they do. So they are not changed and used up in the reaction. I think I told y'all to give the example of you being in healthcare and you getting a lower down payment, right? So that's lowering your activation energy. They don't get used up in the reaction. Okay, so they can be used over and over again. All right, so they're not becoming products, right? They're just sort of the facilitator that makes the thing happen, right? So the enzyme is kind of like the party planner, right? So they're not really there to have fun. They're there to create the party for everybody else, right? So you're creating the conditions to have a good time. So the substrate, these are reactants. They bind to what we call an active site on an enzyme. Okay, so the enzyme, is the active site is where the party happens, right? So that's where the activity is going to happen, the reaction. And so enzymes, some characteristics of them that they are specific. They're going to catalyze just one type of reaction. Okay, they don't, if they do this kind of party, they only do those kind of parties. Like it's only paintball parties or whatever the party is. And then there's another enzyme and they do, clown parties for kids. They're very specific. So saturation limits. All right, so once they become saturated, they don't have any more space to take on anybody else, right? So it's kind of like, hey, only 300 people can get into this club. Once 300 people are in there, nobody else gets in, okay? Somebody's gonna have to leave in order for new people to come in, okay? So regulation. Other chemicals can also regulate that enzyme. So some of them have to be controlled by other things to tell them, hey, this protein or this enzyme can work or no, it can't. Okay. So, yes, there's a party or no, there's not a party. Okay. All right. So first step in this reaction, the substrates, these are your reactants. They got to bind to the active site on the enzyme. Remember the enzyme, is where the action happens, right? And specifically on the active site. So that's the enzyme's role, is to bring the parties together. It's the facilitator. All right. And again, specific, they've got to fit. If they don't fit, they don't get to be a part of the party. Okay. So once bound to the active site, the substrates are going to be held together, making their interaction easier. So the enzyme holds on to them. Okay. They get in that area, it's like, no, you guys are going to have this conversation, right? You've been avoiding each other. It's time for you to have this intervention, right? It's time to have a conversation. You form what we call an enzyme substrate complex, okay? Because it's the enzyme and the substrates. So it's an enzyme substrate complex. Then the substrate that's bound, it's going to change its shape. And this change facilitates creating a new product. Okay, so now you've gone from these two parts to now one thing. Okay, so you've changed it into something else. Now the product is made, it's going to be released from that enzyme. Okay, what happened to the enzyme? It's kind of a trick question. Nothing, right? So all it does is sit there and wait for some more substrates to come by. It keeps, just used over and over again. All right, so cofactors. So what's a cofactor? Well, a cofactor is an ion or molecule, and it's going to bind or connect to some enzyme before the substrates can bind. So remember we said some enzymes have to be controlled by other cellular components? Well, this is one of them, okay? So you got to have a cofactor in order for the enzyme to bind the substrates at the active site. All right, example, so you also have what we call coenzymes, and these are vitamins are an example of these. And so they're organic, which means they have carbon and hydrogen, but they're not proteins. Okay, remember an enzyme's a protein. So these are other materials that are not a protein that bind to the enzyme to make it work. So it's kind of like having, you know, a car, but if you don't have the key, the car doesn't work. But the car has an active site, which is where you put the gas, right? Which is where, you know, any chemical reaction can happen. All right. So proteins have certain conditions that they like to work in. Okay, so it's, you know, they're kind of like the musicians that are like, when I come to your city, I need to have, you know, a bunch of jelly beans in my trailer, right? They have to have a certain temperature. Okay, if temperature is not right, they don't work. Okay, pH is not right, they don't work. Okay, so we call that denaturation. All right, so that means that there's a change in shape and a loss of function because it got too hot, typically, or the pH was too acidic or too basic and damaged it. Okay, because think about temperatures. Temperature can break bonds, and they can break hydrogen bonds. And we saw, remember, with the tertiary and the secondary structure, where you had all those hydrogen bonds in the secondary structure. If you get too hot, you could disrupt some of those, and then the protein doesn't work, okay, because the folding is thrown off. All right, glycoproteins. So glycoproteins just basically means you got a lot of protein, a little bit of carbohydrate. And the way I memorize this is you have a noun and a modifier or an adjective. So protein is the noun, glyco is sort of the modifier or the adjective describing it. So it's mostly protein. It has a little bit of glyco or sugar on it, a little bit of carbohydrate. So enzymes, antibodies, hormones, parts of the plasma membranes have glycoproteins in them. You'll also see glycoproteins in mucus, okay? Now we also have the proteoglycans. So now glycan is the noun. So it's mostly large polysaccharide, some protein, okay? And these chemicals increase viscosity in fluids. So what's viscosity? Anybody got, you've seen like the old oil commercials, they talk about viscosity. Viscosity is thickness, okay? So particularly like if you're thinking about your body fluids, like your blood or other fluids in your body, they have to be a certain thickness, okay? And the proteoglycans give them that, okay? All right, so that's proteins. So based on time, I'm actually gonna go ahead and move on to nucleic acids. We're gonna see how much we can get through so that we can start chapter 3 next class. So nucleic acids. So these are organic molecules in your nucleus or your cells, okay? What do? Well, they store information and they process information, okay? Two main types of nucleic acids, okay? We've got the deoxyribonucleic acid and the ribonucleic acid. Deoxyribonucleic acid is what's determining your inherited characteristics, things you inherit from your parents or down your family tree. DNA also directs the synthesis or creation of proteins, which is why proteins are so important. DNA controls production of enzymes, which should make sense because enzymes are what? What category of organic molecules are the enzymes in? We just covered them. Proteins. and they control metabolism, which means they control the chemical reactions that happen in your body. Now, RNA is an intermediary. It controls a specific step in the middle in protein synthesis. Doesn't control the whole process. All right, so let's talk about nucleic acid structure. So DNA and RNA are going to consist of long chains of nucleotides. All right, so these are monomers. So just like, you have the monosaccharides in carbohydrates and the amino acids in proteins, and nucleic acids, you have nucleotide monomers. And what are they built of? Well, I'm glad you asked. They have a pentose sugar. What does pent mean? Like pentagon. 5, right? So it's 5-membered ring, typically 5 carbons. It could be deoxyribose or ribose. And you also have a phosphate group, remember it has phosphorus in it, and a nitrogenous base. A, G, T, C, or U. That's kind of like the R group in proteins. This is kind of a, something that makes it specific. Okay, it tells you which, it gives you some specificity to the nucleotide. All right. All right, so this is what that looks like. Here's your pentose sugar, 5-membered ring, also 5 carbons, 1, 2, 3, 4, 5. And you have a phosphate group and a nitrogenous base. Nitrogenous base means it has nitrogen in it. Okay. All right, so we classify the nitrogenous bases as purines or pyrimidines. So you have adenine and guanine. Okay. I'm not going to make you memorize the actual structure. What I do want you to know is 2 things. Think of the pu rhymes with 2, because these purines have 2 rings. Okay. The other thing is A and G. And because, I went to Texas A&M, I'm an Aggie, so we used to say the Ags are pure. So that's how we remembered it. But A and G, all right? Or if you want to say Purina, which is an ag product, and it helps you remember, be my guest. All right. And you have the pyrimidines. You have 3 of those. And they all have 1 ring, okay? Cytosine, thymine, and uracil. Thymine is found in DNA only. Uracil is found in RNA only. Okay, I'm not going to make you memorize the chemical structures. Just know A, G, and C are found in both. But thymine is only for DNA. Uracil is only for RNA. All right. So DNA and RNA. All right, so how are they similar? How are they different? Well, DNA has a pair of nucleotide chains. RNA has a single chain. That's the difference. DNA has 2 chains. All right. And RNA has 1 chain. So these 2 chains in DNA are complementary to one another. And these 2 strands are next to each other, and they are held together by hydrogen bonds. Okay. And those bases are held together by hydrogen bonds. And it's not just 2 lines together, but they spin on each other and form a double helix. All right, so now RNA, again, single chain. And there's 3 types: messenger RNA, transfer RNA, ribosomal RNA. And we'll learn about all those in chapter 3. We just want to introduce those to you now. Okay, mRNA, tRNA, rRNA. But remember, DNA, deoxyribose is a sugar. RNA, ribose is a sugar. DNA is double-stranded. RNA is single-stranded. DNA's bases, A, G, C, and T. RNA's bases, A, G, C, and U. Okay. All right. So here's the complementary base pairing rules. So purines are going to pair with pyrimidines. DNA, you're going to see adenine bond with thymine. A binds with T. Cytosine is going to bond with guanine. G binds with C. Okay. A binds with T, C binds with G. And then for RNA, uracil replaces the thymine. Okay. So important to know those rules. A binds with T, C binds with G. All right, so here you can see that. A binds with T, G binds with C. All right, and just like I said, forms a double helix. RNA is gonna have a single chain, has U instead of T. All right, so here's that table I was just talking about. RNA, sugar is ribose, DNA sugar is deoxyribose. So this table, y'all will want to make sure you put this in your memory, okay? Nitrogenous bases. For RNA, adenine, guanine, cytosine, uracil. DNA, adenine, guanine, cytosine, thymine, okay? Typically, the number of nucleotides, fewer than 100 to about 50,000. Deoxyribose, always more than 45 million, so much longer, okay? So much shorter in RNA. The shape varies with the hydrogen bonding. Again, you got three types, mRNA, RNA, and tRNA. DNA is in a double helix, 2 strands, and RNA is 1 strand. Function performs DNA synthesis as directed by DNA for RNA. DNA is going to store genetic information, controlling protein synthesis. Okay, so DNA is the master controller. RNA performs a step that DNA allows it to perform, basically. All right, so real quick, high energy compounds. So most high energy compounds are derived from the nucleotides. So that's why we're covering them here. Phosphorylation just means you're adding a phosphate group to some molecule. And when you add a phosphate group, you're adding a high energy bond. Okay, and you need that for your cells. They need that energy. All right, so the one that you're going to be most familiar with is ATP in this course. Means it's a high energy compound with 3 phosphate groups. If it has 2 phosphate groups, it's diphosphate. If it has 1 phosphate group, it's monophosphate. All right, but adenosine is a derivative of adenine. So that's why we're covering it here, right? It's a nucleotide. And there's an enzyme. Anytime you see ASE, y'all want to write this down. Anytime you see ASE, it's an enzyme, okay? So adenosine triphosphatase, or ATPase, is an enzyme, and it catalyzes conversion of ATP to ADP. So you're breaking that energy bond in ATP to get some energy out for your cells, okay? All right, and this is what this looks like. Here's ATP, so 3 phosphate groups. If you break one, you get ADP. You break another one, you get AMP. All right, but there's your adenine, your ribose. Okay, that's why we're covering here under nucleic acids. All right, don't worry about the chemical structure, just know the parts. Okay, so that's chapter 2. So you can go ahead and do your exit ticket. Those of you who are going to lab, you can do it on the way. On your exit ticket, if you want to add a little bit about nucleic acids, since we didn't get a chance to talk about it, that's fine. All right.